The title for today's lecture, Overcoming the Cancer Ecosystem with Adaptable mRNA Vaccines. Dr. Elias Sayor is a board-certified pediatric oncologist who has served as sponsor of multiple FDA IND submissions, leading to the first in human clinical trials of several compounds. He has spearheaded creation of the University of Florida Pediatric Cancer Immunotherapy Initiative in Large Animal Comparative Oncology Initiative, and he serves as the principal investigator of the RNA Engineering Laboratory, where he has been the primary inventor on a number of pending patent applications. He is also the recipient of an ALSF R-Accelerated Award during an inaugural year in 2022. And with that, I will turn it over to you, Doctor.
Thank you. Thank you so much for the invitation. Can everybody hear me and see my slides?
Yes, we can.
Apologize for the delay. I was freaking out a little bit, but really honored, privileged to be here and to be speaking at this forum. I'm going to be talking about the cancer ecosystem and our work with mRNA vaccines. I do have disclosures. I do serve as a paid consultant for Siren Biotechnology, and we have multiple patent applications on mRNA vaccines with option licensure, and I will be talking about these today. So cancer, I frequently ask our trainees, what is it? And I tend to get a definition of rapidly dividing cells, 2, 4, 8, 16, 32. And I tell our learners that cancer is so much more than that. There's so many unique features to cancer. And rapid cell division doesn't discriminate cancer from a benign tumor that's just growing. So one of the many features of cancer is its heterogeneity. It's not just one population of cancer cells, but actually competing populations of cancer cells. And these cells compete with each other for dominance in an ecosystem that cancer is perpetuating. And this ecosystem is made up of stroma, thick, fibrous tissue, angiogenesis, blood vessels that are feeding these cancers. There's also immune cells. And in many of these tumors, these immune cells are part of the problem. Myeloid-derived suppressor cells, tumor-associated macrophages, regulatory T cells. These all live as part of this environment. And as this environment is being remade by cancer, it's a very difficult environment for normal cells, normal host parenchymal cells, normal immune cells to survive it. These environments tend to be devoid of oxygen or with low pH. And only the most aggressive, only the most evolved tumor cells tend to survive in this environment. And so I really liken, and many people are really likening cancer to more of an ecosystem, making it more of an ecological problem. So why am I showing a picture of the Burmese python? Many might be familiar, but this is an invasive species in Florida, and it's absolutely devastating our ecosystem. This was introduced actually by people who had them as pets and then introduced them in the wild. This is many decades ago. And then this snake really found a home here, loved the environment, and was really naturally selected to survive. And as this snake propagated, it is really remaking the environment. It is dividing very rapidly. And there really has been no solution to this problem. One of the solutions has been a form of quote unquote targeted therapy to really go out and shoot the snake. And that has not really made a difference in its overall numbers. In fact, on a large population scale, the snake is really growing exponentially in terms of its population, even if we're shooting many more hundreds year after year after year. And so I often ask our trainees to try to come up with ways to eradicate this snake. And I've always found it intriguing that many of the ideas that they come up with are therapies that are often being used against cancer, whether it's targeted therapy, differentiation therapy. And I often wonder whether we can just use something as catastrophic as a nuclear bomb to take out the Burmese python. And everyone thinks I'm crazy for suggesting that, and it is. And yet that is how we treat cancer patients, with very aggressive therapies, chemotherapy, radiation, harsh surgical resections. And the reason they work is they are devastating to the environment. They can really allow these cancer cells to die out by basically taking a hold of the environment that they have really grown in. The challenge, of course, is when these malignancies escape. When they do, then it's really only damage that we're introducing. So is there a better path? And one of the challenges with the Burmese python is it's an apex predator. But what if we could introduce a predator to take out these invasive species? And I certainly see cancer as an invasive species that is foreign. And it is the immune system's job to eliminate enemies, foreign and domestic. And the immune system probably eliminates cancers all the time. We just don't give it credit for that because we're blissfully unaware when it's doing its job. Fortunately, when cancer has escaped and has remade the immune system in such a way that it's very tolerant to cancer. And so how can we reset the immune response, reset it in such a way that these environments are being reprogrammed and allowing these environments to facilitate an immune response and not facilitate the cancer? And so here I'm showing data in a tumor. Glioblastoma is the most common malignant brain tumor. And this tumor really is a prototype in the sense that these are very difficult environments. They're hypoxic. There's competing populations of cancer cells. Immune cells have a very difficult time living in this environment. And yet, even in this tumor, which is notoriously heterogeneous and notoriously immunosuppressed, we found that in patients that had more T cells, more effector T cells in the tumor microenvironments, they indeed would have better outcomes. And so this challenges the idea that you either have cold tumors or hot tumors, and immunotherapy only works in hot tumors. And this is even in the natural setting. In the natural setting, you can have an endogenous response in a cold tumor, and that can improve survival benefit. But how can we make these results more frequent and more mainstay? And in many ways, what people have done is they've tried to make cancer vaccines. And cancer vaccines, in many ways, are really the beginning of immunotherapy in the late 1800s, the idea of making vaccines to elicit a response against cancer. And now, nearly 150 years later, we still really don't have a cancer vaccine, a therapeutic cancer vaccine that works to reject cancer. We have immunotherapies, checkpoint inhibitors, CAR T cells, monoclonal antibodies, but still waiting for therapeutic cancer vaccines to kind of meet the promise that launched the entire field of cancer immunotherapy. So why haven't they worked? It's been a lot of testing and a lot of trials. And to make a long story short, it just takes time. Vaccines really require many months, years really to boost an underlying response. And this is evidenced by any primary vaccination series in children. And so those primary vaccination series often require months, years of boosts to get protective immunity. And that is in the immunocompetent setting. In cancer, responses need to be induced much more rapidly against an evolving enemy. And that response has to work extraordinarily rapidly to basically not allow the cancer to get away. And so if we are to make a cancer vaccine, it has to elicit responses, I would say, not over weeks or months, but really in hours, days. This response should take hold against cancer. And mRNA really does provide an opportunity. The Nobel Prize was recently awarded to the original discoveries that paved the way for mRNA vaccines in the COVID-19 pandemic. And many people are trying to follow the COVID-19 blueprint to create mRNA vaccines against cancer, including Moderna and BioNTech. And this paradigm is largely based on taking a gene of interest. In the case of COVID-19, it's the SARS-CoV full-length spike protein, and encoding that as a messenger RNA. RNA is very unstable, and so you need a lipid particle shell to protect that RNA from degradation, and then it can be delivered to patients. At least the COVID vaccine is an intramuscular injection to induce a response. Now, the advantage of RNA, and I think we've seen this on the COVID-19 scale, is you can adapt it. And I think that is a huge attraction to mRNA. I remember the CEO of BioNTech was on the news, and they said, the reason we've never had a coronavirus vaccine is because the virus is constantly changing. It's constantly adapting, very much like cancer on a large scale. And he was very matter of fact in saying that mRNA can be adapted with the pandemic to go after new variants. And we're seeing that play out as new vaccines, new boosters are made to account for the heterogeneity of the SARS-CoV-2 virus. And so could we use a similar paradigm to treat cancer in a human being where the RNA can be adapted? And I think the answer is yes, but we also can't forget that many of the challenges that were salient for all cancer vaccines, peptide vaccines, viral vaccines, nucleic acid vaccines, et cetera, those challenges exist as much for RNA as they existed for any of those platforms. And that is, even if you can get the best T-cell response in the world against your tumor, and even if it's adapted against the most resistant variants or the newest antigens present in one's cancer, those T cells still have to survive in these very difficult environments, these notoriously heterogeneous and immunosuppressive microenvironments. And so that is an extraordinary challenge for mRNA, as it is any vaccine. And so one paradigm that we have been working on to enhance mRNA vaccines for many years now is really by just making it look like a very dangerous virus. And the virus that induces response most rapidly, to my knowledge, is influenza. Influenza, from the time of infection to the time of symptoms, where you're acutely symptomatic, is something like 2 days, 2-3 days. Very rapid infection from the time of inoculation to symptoms. And those symptoms are largely mediated by the immune response. Your innate immune response kicks in very rapidly, and that innate immunity is just buying time for your body to make a specific response against influenza and get it under control. And what is influenza? It's single-stranded RNA in a nanoparticle shell. And when that hits your bloodstream, that elicits a very significant inflammatory response. And can we use what nature has already given us in terms of recognizing particulate matter in the bloodstream? Can we harness these mechanisms that already exist in all of us to now elicit strong immunologic responses against cancer? And so over the last 10 years plus, we have worked on making nanoparticles, different types of nanoparticles, around 100 to 200 nanometers in size. Many of these RNA nanoparticles are shown here, with the intention of introducing them into the bloodstream as an intravenous administration. So unlike the COVID-19 vaccine, which is given locally, this is given straight into the bloodstream to mimic viremia and elicit very rapid responses in a matter of hours. Now, over the last few years, we've refined this approach. There have been papers that have shown that viruses are more immunogenic if they're clustering, meaning if you have single viral particles and they're infecting different cells, that may not be as immunogenic as clusters of viruses, viral particles infecting cells. And so we've tried to leverage that understanding to make these multilamellar lipid particle aggregates, where we can basically package more RNA into a particular payload. And at the end of the day, RNA is just information. Whether it's spike protein or mRNA encoding for cancer antigens, it's just information designed to alert the immune system. The difference between COVID or spike and cancer antigens, though, is spike is foreign. When spike is expressed, that elicits a massive immunologic response. Cancer antigens are already expressed, and the immune system does not recognize that as dangerous. When a cancer is manifested, the immune system sees it as self, and it shouldn't. Now, I would argue that a virally infected cell is more like self than a cancer cell. A virally infected cell is 99.9% self, and yet the immune system knows to kill it. And yet, for a cancer cell, which I would argue is far more foreign appearing, the immune system has become very, very tolerant to it. And so for us to really unlock immunotherapy, certainly vaccine immunotherapy against cancer, we have to have designs that look really, really dangerous. And this particular approach, this lipid particle aggregate design, is really to do just that, to make this payload look very dangerous to the immune system. And so here are some pictures of many of these particles. These empty particles are these are bilamellar lipid particles. And when we formulate them as multilamellar aggregates, you can see they form these onion-like structures, and these kind of stick to each other, aggregate. You can see that better here, that these tend to aggregate, and this is zooming out a little further. And unlike our previous RNA nanoparticle designs, these are a bit larger, around 200 to 400 nanometers in size. So bigger than our previous particles, but still small enough that they should be filtered out within the lymphoreticular system after an intravenous injection. And within the lymphoreticular system, lymph nodes, spleen, liver, they can induce immunologic responses. So I've spent some time talking about the particle design itself, or new particle designs we need to unlock immunotherapy against the cancer ecosystem. But what about the RNA itself? So we have pursued two different approaches, a personalized approach to vaccine immunotherapy and an off-the-shelf vaccine approach. The personalized approach can be obtained from very few amounts of tumor tissue, from 100 to 200 cells of a biopsied cancer. Our total RNA can be extracted. This RNA represents the ribosomal, the transfer, and the messenger RNA components. This can then be made into a cDNA library by RT-PCR. This cDNA library now is the fingerprint for that patient, the fingerprint of their tumor-specific transcriptome. And then this can actually act as a library that we amplify mRNA whenever we want for loading into a lipid particle vaccine. And this is renewable. We can amplify mRNA for continued vaccines, continued boosts. And the idea of these personalized vaccines are to localize in vivo to immune cells for activation of a T-cell response. Now, the other opportunity mRNA presents is we can make off-the-shelf vaccines. And these off-the-shelf vaccines, particularly for pediatrics, can be designed from an optimized backbone where a target gene of interest can be introduced into that backbone. So for pediatric cancer, one could imagine there are many, many targets that could be utilized. There could be fusions that lead to new peptides, epitopes that can be preferentially targeted as an optimized backbone. This could certainly be quite salient for Ewing sarcoma, rhabdomyosarcoma, ependymoma, which are often fusion-driven. And this could also be leveraged for common mutations across pediatric cancers, whether it's a BRAF mutation, the dreaded H3K27M mutation in DIPG. But basically, this allows you to plug and play, identify these constructs, clone them into these backbones, deliver them as off-the-shelf vaccines. And these backbones are really the intellectual property that Moderna and BioNTech have used to develop the COVID-19 vaccines. So what we found is when we inject these multilamellar RNA lipid particles encoding for either personalized or off-the-shelf targets, they go everywhere. And this is true of a virus. A virus doesn't say, I want to go to your dendritic cell sitting in your spleen. They tend to go everywhere. Now, the spleen, as many of the lymphoid organs in the body, are designed to be to pick up particulate matter much better than many of our other organs. And so these RNA nanoparticles are not designed to, with the targeting moiety for a specific cell or organ, but they do get taken up in what I would refer to as the organs of interest: liver, spleen, lymph nodes, and as the filtration system of the body. And they do localize to a small degree to tumors, even brain tumors, because these tumors tend to have leaky blood vessels. These leaky vessels are part of lymphoreticular organs, but the tumor can also disrupt blood-brain barrier and vascularity to such a degree that particles can also localize there. And we found that, to our surprise, these newer, larger particles didn't get into dendritic cells as well as we had hoped, but they actually localize more to stromal cells. You see these elongated cell shapes over here. These have been found to be fibroblastic reticular cells. And so these stromal cells are actually not useless cells. And even though they're not the specialized immune cells that we're interested in, these fibroblastic reticular cells co-localize with more prototypical immune cells, cells like macrophages, which are seen here. So they actually can directly contact these immune cells. And it's been shown across the literature that fibroblastic reticular cells can actually shuttle antigens into antigen-presenting cells like macrophages, dendritic cells. So we think these cells are actually very important in inducing a cytokine response, and fibroblasts are very important in inducing type 1 interferons. And we also think they're very important in helping mediate an adaptive response through shuttling of antigen to macrophages, dendritic cells. And we also found that they can actually just release protein into the local regional milieu. And this was shown here in vitro, where we transfected a human fibroblastic reticular cell line with GFP RNA nanoparticles. And as you can see here, there are actually very few cells that are expressing the GFP. Most of the GFP was actually in the supernatants, basically being released by these fibroblasts. So these stromal cells are very important. They're important for helping catalyze an innate and we think an adaptive response. We're trying to understand this better. And stromal cells are also part of the weed, the ecosystem of the tumor, that if we can reprogram it, our hope is that we will create more pro-inflammatory microenvironments. So as soon as we administer these vaccines into the bloodstream of animals, we see very significant cytokine, chemokine responses. There is a surge of type 1 interferons, both interferon alpha and interferon beta. There's also a surge of type 2 interferon, interferon gamma, TNF alpha, IL-12. There's also a significant increase in chemokines like CCL2, CCL4, CXCL9, CXCL10. These chemokines are well-known recruiters of both myeloid cells and lymphoid cells. So we're getting cytokines that activate the immune response. That's shown here on the left. And then we're getting chemokines that mediate trafficking of those immune cells to basically moving those immune cells to the lymphoreticular organs. And that's shown here, that within 6 hours of an RNA nanoparticle injection into a mouse, we see rapid mobilization of monocytes. Basically, monocytes are nearly undetectable after 6 hours. 6 hours after administration, they're trafficking to lymphoid organs. And the lymphocytes also do the same thing. They're trafficking to these lymphoreticular organs. And simultaneously, if we were to look in the spleen, we see significantly increased percentages of activated dendritic cells and significantly increased numbers of both CD4 and CD8 T cells. So What I'm showing here is really the innate response that very rapidly, after one systemic IV vaccine, you are seeing a surge of innate immunity, a rapid surge of innate immunity. What I haven't shown you, but is true, is that surge is very transient. Within 24 hours, those monocytes recover, those lymphocytes recover, and the cytokines, chemokines largely defervesce, which is important because you don't want to sustain innate immunity for very long. Sustained innate immunity can lead to chronic inflammation. Sustained innate immunity can lead to actually regulatory T cells. And the reason these surges in innate immunity are important is because this can help an adaptive response. Adaptive immunity, as mentioned, takes time, and these have to be boosted. But through these innate surges, if we are able to reprogram the tumor microenvironments, if we're able to reprogram systemic immunosuppression, now this adaptive response has a fighting chance. And sure enough, if we were to look at even the brain tumors of mice that were given one vaccine, and look within these brain tumors 24 hours after a single injection, we see significant reprogramming in these tumors. Genes for antigen presentation, RIG-I-like receptors or pathogen recognition receptors, interferon signaling, cytotoxicity, DNA damage, these are all upregulated within 24 hours of a single vaccine. In effect, making these microenvironments hot, more pro-inflammatory, more conciliatory towards an immune response or an adaptive immune response that you're simultaneously activating. And on the left, we can polarize significant adaptive immunity through repeated mRNA vaccines, but these T cells need an environment that allows them to survive. They need an environment that allows them to induce their effector functions so that the response can continue to proliferate and be maximized. In terms of anti-tumor efficacy, we've looked at a plethora of preclinical models. This includes both pediatric and adult models, and we've seen significant anti-tumor efficacy across these. In a preclinical model for osteosarcoma, we showed that personalized vaccines derived from the K7M2 cell line induced significant response. In the KR158B model of glioblastoma, this is a mouse model for adult GBM. We showed that PP65 or tumor-specific off-the-shelf vaccines were superior to control vaccines. And then, most importantly, in a brain tumor model for DIPG, in these models, vaccines were introduced fairly quickly to see if we can ward off a minimal residual disease setting. As many know here, DIPG is not resectable. This is not an MRD setting. We started vaccines at day 35 when animals became symptomatic, and were able to elicit significantly improved survivorship, long-term survivors in this model imported to our laboratory from Dr. Becker. And this model expresses the H3K27M tumor, and mice were vaccinated with H3K27M specific vaccines. Mechanistically, there are a lot of cytokines, chemokines that are upregulated to both activate the immune response, mobilize the immune response, but critically important is the type 1 interferon response. The type 1 interferon response is like the 911 phone call. In the apps, it alerts the immune system. It alerts all levers of the immune system that there is an infection. There is something dangerous that is present. And sure enough, if we block this type 1 interferon response with a receptor blocking antibody, we abrogate the anti-tumor efficacy from our vaccine. As we tried to understand this mechanistically, we just assumed, because BioNTech had published this, that single-stranded mRNA vaccines, at least their cancer vaccines, work through toll-like receptor 7 activation. Toll-like receptor 7 is basically our body's way of recognizing single-stranded RNA as foreign, but In knockout mice for TLR7, we still got some pretty good effects with our vaccine, and we also got a really strong type 1 interferon response. And this was a bit of a surprise that because we were using single-stranded RNA, and yet mechanistically this was not dependent on a TLR7 response. We did take it one step further. We looked in knockout mice for MyD88. MyD88 is the adapter protein downstream of TLR7, and MyD88 is also the adapter protein for other toll-like receptors. So we thought that if we knocked out this adapter protein, that we would also abrogate response. And sure enough, we were still able to have significant anti-tumor efficacy, suggesting that these responses are not entirely dependent on TLR7 or MyD88 signaling. But to our surprise, it was actually RIG-I. RIG-I is a sensor. It's really more for double-stranded nucleic acids or double-stranded RNA. RIG-I is a sensor in stromal cells, is a sensor in fibroblasts, and this in knockout mice or RIG-I knockout mice, we were able to significantly attenuate the type 1 interferon response. We were also able to abrogate long-term survivorship in these knockout mice relative to wild-type mice treated with our vaccines, demonstrating that RIG-I is critical for the immunologic responses seen. It's not entirely dependent on RIG-I. We're still seeing some interferon, and we're also seeing still some survivorship. So we're likely activating multiple pathogen recognition receptors, but RIG-I is clearly very important in mediating much of this response. So in terms of our schema for response, we think these RNA lipid particles, these multilamellar aggregates, localize all over the human body, largely to lymphoreticular organs, which have stromal cells that pick up these payloads. These fibroblasts can release copious amounts of type 1 interferons, which induces a damage response, also releases cytokines, chemokines that elicit mobilization, massive mobilization of monocytes, T lymphocytes. And once they localize to this environment, they can be fully licensed through antigen capture. And these dendritic cells can communicate or talk to T cells, activate them against these cognate antigens, and these T cells can now traffic across the human body and survive, importantly now survive in the tumor microenvironment that's being simultaneously reprogrammed, exert their effector functions, mediate rejection of these malignancies. RNA vaccines have tremendous capacity for enhancement. So in many ways, what I've shown here is that we think of them as vaccines with the idea that they can induce an antigen-specific response. I think of them as immunomodulating agents as much as I think of them as vaccines, but they also can be leveraged to package multiple payloads. We showed many years ago that you could package tumor antigens and RNA encoding for immunomodulatory cytokines to enhance the immune response. And so these approaches allow you to package many different payloads. It can be not just tumor antigens, but immunomodulatory cytokines. It could be even for information that's encoding for a monoclonal antibody to target a resistance mechanism within the tumor. Could be PD-1, could be another checkpoint, could be IDO. You could also use siRNA or small interfering RNAs, co-package these with RNA nanoparticles, induce enhanced immunity. This was shown by Dr. Grippen, a graduate student in the GL261 model. You can also use these vaccines in combination with other therapies. So in partnership with Dr. Huang, who's developed CAR T cells within our group, and Dr. Castillo, they showed that you could actually combine these things for synergistic effect, that CD70 specific RNA nanoparticles or vaccines encoding for the surface antigen of interest can substantially enhance the long-term survivorship induced by CAR T cells. Similarly, in partnership with Dr. Silver, we were able to combine these vaccines with checkpoints and showed that we could get tremendous synergy in a head and neck cancer model. So these vaccines are not just immunomodulatory agents or prototypical vaccines, but can be leveraged through intelligent designs of the backbone, intelligent designs of the nanoparticle or payload development, and through combination therapies can be designed to create new regimens that can cure these distinct cancers. We did look at toxicity in preclinical models. We have done comprehensive GLP tox testing across mice. I can confidently say that there aren't significant late effects. At 35 days, 56 days, at 110 days, there weren't significant late effects. These mice had normal organs relative to control animals. Like many immunotherapies, it's the early effects that we have to be mindful of. The early effects of cytokine release syndrome, the early effects of mobilizing the immune system in such a way that could lead to side effects. But there were not any gross or microscopic test article related findings at later time points. And this was an important foundation really for moving this forward into human clinical application. And the FDA often wants to see not just small animal data, but large animal data. And here at the University of Florida, we've been able to leverage our partnership with our College of Veterinary Medicine to take development of vaccines and new vaccine designs and test them in large animals. And large animals have the pediatric forms of cancer. Osteosarcoma in a dog is nearly 100% homologous to the human form of disease. What kills a dog with osteosarcoma is the pulmonary lung mets, which is similar to what happens in patients when osteosarcoma breaks through. And glioma is very similar. Glioma in a dog, canine glioma, recapitulates the pediatric version of high-grade glioma more than it does glioblastoma in adults. We've worked with Dr. Carrera-Eustace, Dr. Milner, Dr. Souza. We have trials now in dogs with glioma, dogs with osteosarcoma, and actually cats with Dr. Souza, cats with head and neck cancer. I'm going to show some data in our dogs with glioma. I should make sure this is clear and highlighted. These are pets with spontaneous, natural forms of cancer. They're enrolled on a trial through their owner's consent. These patients have diseases that are incurable. And if their owners are willing for them to go on investigational therapy to improve their outcomes through consent, we can do that. And so, and we pay for everything. And so these animals were, with glioma, we stratified animals to two groups, Group A or Group B. Animals across both groups got 3 vaccines 2 weeks apart. However, in the Group B cohort, we gave a neoadjuvant vaccine, a non-specific neoadjuvant vaccine to test what, this was before biopsy, to test whether indeed we could reprogram these tumor microenvironments, which I will show you. But in all of these dogs, we saw increases in type 1 interferons, mobilization of chemokines. Many of these patients had flu-like illnesses, as demonstrated through low-grade temperatures. And we saw rapid mobilization of monocytes and lymphocytes, similar to what we saw in the mouse. Now, truth be told, it was this observation in the dog that this is the first time we saw the massive mobilization of monocytes and lymphocytes. It was actually this result in the dog that made us go back to the mouse and prove that this was happening. And in the dog, you can see that the kinetics are interesting. Monocytes nadir at 2 hours. So right after a vaccine, 2 hours later, monocytes drop near 0. And lymphocytes trail, and they nadir at around 6 hours. So these particles get out of circulation very, very quickly. And monocytes are being recruited to pick up these antigens. And then lymphocytes are being recruited to have these antigens presented to. And then they can be active and can find these tumors and hopefully kill them. Now, We did images on these dogs before vaccines, after vaccines, and what we noticed across many of these patients, this is the first patient shown here, is that they would develop decreased enhancement, but more swelling in the tumor. And in fact, the patients who looked like they had the bigger tumors or more swollen tumors actually had better outcomes. And we think this is evidence of pseudoprogression. We didn't do serial imaging on these patients because we couldn't afford to do serial imaging, but we didn't do it, so I can't know for sure if these patients develop true pseudoprogression. But across this cohort of 10 animals, the median survivorship was 123 days, and some of our best responders certainly had this evidence of sometimes doubling in their tumor size. And this 123-day median survivorship, again, was in animals who just received vaccine therapy. We have one animal here shown with a cross that received chemo before, and one animal shown with a dash here that received radiation afterwards, but every one of these patients otherwise would have just received vaccine alone. And shown here on the left is the reprogramming of the tumor microenvironment in these dogs. And so these Group B dogs got a vaccine 24 hours before biopsy. They were compared to the Group A animals who received no treatment before biopsy. And you can see that these microenvironments were reprogrammed more or less in favor of genes for antigen presentation, interferon signaling, cytotoxicity, and DNA damage. So this was critical, the large animal data, to obtaining an FDA IND, to running human clinical trials. We wanted to run a pediatric trial first, but the FDA really mandated a cohort of adult glioblastoma patients before we could test this in children. And so for adults with MGMT unmethylated glioblastoma, tissue was obtained from a biopsy for manufacturing of personalized vaccines that were mixed 1-to-1 with PP65, which is a tumor-associated antigen in both adult and pediatric high-grade gliomas. And then patients received biweekly vaccines and then monthly vaccines. And so I'm showing data here for the first 3 patients. In the first 3 patients that we treated, we saw significant increases in inflammatory cytokines like type 1 interferon, TNF-alpha. These were very quick. We would notice them within the first couple of hours, or at hour 2, they were increased. And then it wasn't until after hour 2 that we saw these secondary cytokines, more demonstrative of an adaptive response, type 2 interferon, IL-12, IL-15. We saw after 2 hours. There was also a significant increase in chemokines over these time points. So again, activation of immune cells with cytokines, recruitment of immune cells with chemokines, but CXCL1, CXCL2, 3, 4, 5, CXCL 9, 10, 11 were all increased after vaccination. And this correlated, as shown in mouse and dog, with margination of monocytes, so natering of monocytes at hour 2, natering of lymphocytes by hour 6. And then what we didn't see in our animals was this surge in neutrophils, significant increase in neutrophils at 6 hours, which defervesced in the majority of patients by 24 hours. We did more comprehensive flow cytometry looking at dendritic cells. Dendritic cells also marginated, as shown in mouse. They disappeared from peripheral blood. But when we fractionated this in terms of the myeloid-derived dendritic cells, which are your prototypical antigen-presenting cells, and plasmacytoid dendritic cells, which are really more of innate cell types that secrete type 1 interferons, you have the myeloid cells seem to be marginating, and the plasmacytoid dendritic cells actually increase in the peripheral blood, mediating much of this innate immune response that we're showing here. And then T cells. Their activation profile was assessed through the CD69 marker and didn't change at hour 2, but then was significantly increased by hour 6. So again, correlating with the kinetics of this response, there's an early type 1 interferon response by 2 hours. Monocytes are marginating then, and then you're getting more of a type 2 response, T cells that are being activated and mobilized to these lymphoid organs. In terms of antigen-specific immunity, we did look for response against PP65, which is a human-relevant GBM antigen. We did see expansion of antigen-specific T cells to this antigen in our patients, and this is shown here by tetramer, also shown by TCR-V beta analysis, and through ELISpot here. And then in the last patient we've treated, they developed significant enhancement after the fourth vaccine, significant enhancement, and was thought to have really tumor progression, or what we thought was a massive tumor progression. That patient had a full resection of this enhancing lesion, and there were no viable tumor cells. And what's shown here are actually immune cells, or CD8 T cells, that were significantly increased post-treatment. So I want to really take time to acknowledge Alex's Lemonade Stand. Alex's Lemonade Stand is funding the work that's allowing us to now move from our preclinical studies and large animal studies and adult patients into children through a multi-institutional consortia. But Alex's Lemonade Stand also, I'm not sure they're aware, but they actually awarded the first grant for this work over 15 years ago to my mentor, which Dwayne Mitchell, which allowed me actually to be recruited into the lab and start working on this project, which I have now carried forward post-fellowship. And this has led to additional funding and hopefully additional trials that will benefit patients through these consortia, both using personalized vaccines for pediatric high-grade gliomas and off-the-shelf vaccines such as H3K27M or DIPG. So in summary, we've pioneered novel formulations that challenge existing paradigms, such as the need for packaging RNA into the core of a lipid particle. We've challenged paradigms such as the need for toll-like receptor-dependent activity, and also paradigms that essentially mandate that we have to localize these particles to dendritic cells. Through multilamellar formulation, RIG-I activation, and intravenous administration, these particles actually allow dendritic cells to traffic to them, as opposed to particles to the dendritic cells. And we think this can be, if safe and hopefully early signs of effectivity, we think it can be adapted for combination therapy and enhanced response that will hopefully lead to new standards of care for patients with these awful tumors. So I'll stop there. I apologize for starting later, and if there are any questions, happy to take them.
Okay, thank you very much. That was very interesting. And we do have a bunch of questions that came in, so I'm going to jump right in if you're ready.
Sure, please.
Okay. The first question says, can targeted vaccines only be made from fresh tissue, or FFPE, I'm not sure what that means, / prior extracted tumor DNA may be able to be used also?
So that's a great question. Generally speaking, the FFPE is basically formaldehyde fixed tissue. You could still do it because there's probably still RNA there to make a vaccine. It's just probably not as representative as fresh tissue that is fresh frozen, because that's more representative. All the RNA should hopefully be there if it's preserved correctly. So you can still make a vaccine, but it is not going to be as representative. It's not going to be as good. Okay.
Next question that came in says, can you please shed some light on the immunogenic mouse model used in the study? Are these mice expressing oncogenes, or are they xerograft models?
Yeah, so I showed several models. The K7M2 is a cell line. It's a cell line for osteosarcoma that was just inoculated into biopsy mice through intravenous administration. And then we basically let that grow and then vaccinated the mice. The KR158B model is an NF1 P53 knockout model that was developed as a GEM model at Tyler Jacks's lab in MIT, and has since, the cell line has been made from that, and that cell line is what we implant into adult mice. Again, recapitulates adult GBM, and then are vaccinated. The other efficacy data I showed was in the DIPG model. This is a model that has, it's not 100%. In our hands, it's about 1 to 10% H3K27M expressing, but has the histone 3 K27M mutation. Again, this was developed at Dr. Oren Becker's lab, and a cell line was made from that model, and that's the K2 line. And that's implanted into neonatal mice. It's allowed to grow for 30 to 40 days, and then we vaccinate those animals.
Okay. All right. The next question says, thank you for the talk. Fantastic work. I'm curious to learn the strategies you implemented to define mRNA specific to tumors. Also, have you tried therapeutic settings to see the efficacy in effector cells responsible?
Yeah, so great questions. Hopefully I can remember all of that. So right now, for the personalized vaccines, we're just using unfiltered RNA. And so that's why it's hard to identify the tumor-specific responses outside of PP65, which is co-delivered. There are ways of doing that, of course. If we could predict neoantigens, clone those neoantigens into cells or their TCRs and show recognition. We're also looking at actually doing this in a longitudinal manner where we can predict neoantigens from patients, make mRNAs encoding for them, and give vaccines that can allow us actually to track response to personalized antigens, but haven't done that yet. I think the second part of that question was the immunologic response that we were inducing and characterizing. What I can say is it seemed if we were to look in the tumor microenvironment, central memory T cells are critical for response. So taking our data, we need central memory T cells, and we also need activated dendritic cells. The two of those things were the things that most strongly correlated with long-term survivorship and the ability of animals to survive re-challenge.
Okay, the next question is, how does the brain anatomy of dogs compare to humans? And does the tumor microenvironment of dogs mimic that of a developing human brain?
That's a great question. I don't know that I'm going to be able to answer that question as adequately as I should, not being a veterinarian. What I can say is there is a cancer cell paper. It was published in the last few years, an excellent paper that really did a deep dive into canine brain tumors. Deep dive. They showed the mutations, they showed the pathways, they defined the microenvironment, if I'm recalling correctly. I can't remember the authors, but it was in the last few years. It was in Cancer Cell. And interestingly, they found that this very much correlates with the pediatric brain tumors. So based on that paper, I would say yes, this is a pretty faithful model to pediatric brain tumors. Oh, you're on mute.
Sorry, I do that once every meeting. There's one more question, and it's from Ingo. Hi, Ingo. How many targets from the malignant transcription do you load into the nanoparticles? Is it indiscriminate? And maybe he said, maybe he missed that.
So it is indiscriminate. This comes up probably in every talk I give because there are a lot of, you can imagine there are a lot of normal antigens because it's indiscriminate. What I can say is anything that is autoreactive should have been eliminated. Any TCR that is autoreactive against presented epitopes that are self, those TCRs shouldn't exist. And really all that's left should be TCRs that recognize foreign material that's presented. Now, I acknowledge that there is a risk of breakthrough immunity against self and autoimmunity. And we are trying to really, that's one of the reasons we're pursuing the neoantigen approach. That's one of the reasons we are pursuing a more filtered approach, because you can actually filter this also through subtractive hybridization and other means. But most importantly now is to at least identify a safe starting dose, effectivity in patients as we refine, iterate, combine, and ultimately, but I would say that yes, this is unfiltered right now.